Ah, let's see here. Trying to use this audio.
Okay. I'll just show you the document that I prepared. And look, I got a virtual background. I'm in space. But I just thought there was a few things that you guys would want to focus on your studies for this topic. So I'm showing my, oh, is my iPad connected? It is, right? What's going on here? Oh, I guess it didn't connect somehow. Sorry, 1 second. There we go. Now can you see my iPad screen?
Yes.
Okay. So let me know if anyone else tries to join, but I'll just tell you, I thought that in chapter 3.1, there's a couple things to, don't forget about the sickle cell disease. They love to do questions on sickle cell. So don't forget about that. I put all the information in here that you would need. And then I put in everything from chapters 3 and 10 do meiosis. And honestly, it's not that different. 10 goes into slightly more detail, but not very much more detail. So I put in the chapter 3 stuff first, but if I was you guys, I'd just kind of skim over that, because then I have the chapter 10 stuff down here. Ooh, my screen just went black. I don't know why. Oh, there we go. Now it's back. And I've got an animation here that's good that you can watch. And the chapter 10 stuff is basically the same. It goes into a little more detail on the difference between chromatids, homologous chromosomes, sister chromatids, centromeres. Those are tough terms. So I'd go back and review those terms. That was you guys.
Yeah. Different.
Yeah. I mean, those are just easy. By the way, can you hear me better today?
Yeah, I was able to hear you just fine yesterday too.
Okay, I'm trying using the headphones as the microphone instead of the computer screen and seeing if it's different. But it's the same basically.
Yeah, I think it's the same.
Okay, just curious. So I'd go over that, crossing over. There's a good diagram here. Independent assortment. There's these pictures, hopefully you get that multiple choice question where it's like number the chiasmata and you just have to count the X's. That's a really easy question.
So for that one, it would be 1, 2, 3, 5?
Yep, 5.
Okay.
I mean, those are about as easy as it gets on the IB test. And it breaks this down into steps. We went over this pretty recently, so I don't know, because we just did meiosis reproduction. I don't feel like we need to go into a ton of detail in this review, but I would go over this stuff for sure and make sure that it all makes sense to me. And then I would try to explain it to somebody else if I was studying for this test. Because I don't know if I want to take the time. We just barely went over this. I don't know if it's worth it for me to write everything out again and re-outline it. I might just try to explain it to somebody and see if I understand it well enough to explain it. And if I can explain it, I probably am prepared for the test.
Okay.
Yeah, so there's independent assortment. So this is really chapter 3 and chapter 10. And it talks about genetic variation. There's good diagrams here.
Hey, Larson?
Yeah.
So is the chiasmata the crossing over?
Yeah, so when they cross over, so like in this picture right here, the chiasmata would be right.
Okay. And then independent assortment is after they go through meiosis completely and separate into the 4 different cells.
Yeah, so in anaphase, they're pulled apart, right?
Okay.
In anaphase, they go through independent assortment. And that happens in anaphase 1 and anaphase 2.
Okay, yeah, that makes sense because there's, yeah.
Explain why this is a reduction division. This explains it really well here, I think. Where is that? I actually think it's down here. Oh no, it's up here in anaphase 1. But I would just go over meiosis. I mean, do you have specific questions?
Not really. I think, remember how we did those practice problems?
Yeah.
Like we just took a day, did those practice problems. Do you think you could post more of those with the answers so that we could practice at home?
Were
Or just
those,
posting
what
same
kind
one?
of practice problems? Remind me specifically about the practice problems.
So I think it had a lot to do with, like, at first it was easier and it started with the Punnett squares. And then as it incorporated like meiosis or the
Yeah.
sex link stuff.
Okay, yeah. Just kind of like some of those worksheets almost.
And the answers.
Because yeah, genetics is like the one topic in biology where you have like worksheets and stuff. So yeah, I can post some of those with the answers.
Thank you.
And you can practice, doing Punnett squares. Look at those predicted ratios for the dihybrid crosses in chapter 10. That's something that I think a lot of people missed. And it just basically tells you if you don't get the Mendelian expected ratio, then the genes are probably linked. If you get the Mendelian expected ratio, then they're probably unlinked because Mendel didn't know about linked genes.
What's the Mendelian expected ratio? Is it the 1-1-1-1 ratio?
So it can be that. It could also be if you do like a dihybrid cross, so if there's like crossed with this, you would get the 9-3-3-1 ratio. But if you crossed, if it was like homozygous dominant, homozygous recessive, then it's like 1-1-1-1. So it just depends on the cross.
Okay.
But you don't have to memorize very many of those. And just realize that if they give you a question and you're looking for a proportion of offspring to be a specific way, they're either going to meet the ratios that you expect, or they're not going to meet the ratios that you expect. And if they don't meet the ratios you expect, then that's because the genes are linked.
Okay. Yeah. Got it.
Polygenic traits, you guys were good on that. Non-disjunction, you guys were good on that as far as I remember. You guys were pretty good at genetics, if I remember correctly, for the most part. The one thing that I think people miss questions on, which is kind of a memorization thing, is some of this stuff down here is this biotechnology stuff. So
yeah,
so I put all the biotechnology stuff into this study guide. And I would go over this. The gel electrophoresis, that one you guys were pretty good on actually.
Yeah.
And PCR. PCR you're pretty good on as well. But then if you keep going, then there's the DNA fingerprinting. It's not very hard. And people miss those questions sometimes. There's the DNA profiling, the genetic modification. Make sure that you go over this right here.
Okay.
That's an important thing to go over. And just make a flashcard, study that. Cloning, very similar to what we just talked about with genetic modification. It's just the whole genome instead of parts of genes. Don't forget about binary fission. That's how prokaryotes reproduce, and budding, that's more of a yeast type of thing. I wouldn't study those a ton, but I would just don't forget about them.
Okay.
And then I put in this stuff. Let's see, what's this one right here? Yeah, this is more cloning stuff. This is the chapter 3 and the chapter 10 cloning stuff. So I would go, oh, this is the other one. This is the plasmids.
Oh yeah, I remember plasmids.
Yeah,
They're
this
kind
is
of similar to the biotechnology thing.
what?
These are kind of similar to the ones you showed me earlier, like the genes transfers.
Yeah, very similar.
Mhm.
And I think if you study them,
Oh wait, this is gene
you know,
transfer.
I don't think it's something that you have to spend a lot of time on. But if you just review them a couple times, I think you'd probably be good.
Yeah, I was good on this.
And then there's a little bit about the possible benefits and risks of GMOs. But I kind of thought this stuff would be the more important stuff, the biotechnology stuff. And then at the very end of this review is the gene pool stuff, where ratios of alleles will change over time based upon, you know, different driving evolutionary factors.
Okay. Yeah.
So that's kind of what I prepared for the review. I'm not surprised that this one is more sparsely populated than the last one. But I just take, no one actually asked any specific questions prior to this one. So I didn't even know if anybody would log on. But do you have any specific questions that you would like to go over, or anything? I mean, it doesn't even have to be limited to this topic, since you're the only one that's here.
Not at the moment. I think your review guides really help. Like as you go through at the beginning of every session, like a review session, you basically answer all the questions that I have. And so I just have to go over the review guide myself and then just study it a bit more. But yeah, I think you gave us all the information. Thank you.
All right, well, I'll do the same thing tomorrow. Even if nobody shows up, I'll post a review guide and I'll kind of say what I think is important about it. And then I'll post that video. It takes a while to process these videos, so they get posted later in the day. But I can do the same thing tomorrow.
Yeah, thank you.
All right, no problem. Well, let's end this then.
Okay.
Okay, see ya.
Bye.